Good morning, dear students. I'm glad to see you on my lecture. I hope that all are here and we can start to work. Please listen to the lecture, look to the pictures, and also maybe do some conspects, because in explanation I'm also telling about important things. Let's start. There is an ancient Greek myth about Titanos Prometheus, a culture hero who was credited with creating people from the clay. He stole fire from the Olympic gods and gave it to the people as a gift for their development and progress. Zeus, the king of Olympic gods, was very angry and decided to punish him, and immortal Prometheus was bound to the rock, and each day an eagle was sent to feed on his liver, which would then grow back overnight to be eaten again and again next day. The ancient Greeks often thought that the liver is the seat of human emotions. Any case, even our ancient ancestors understood the importance of this organ for the life and its regenerative properties. In mythology, over the time, Prometheus was freed by Hercules. In reality, you will save the livers and lives of your patients with yourself. So, today's topic of our lecture is biochemistry of liver. Please, small notes. It is more convenient for me to give a lecture as a whole without interrupting for the breaks. So therefore, at any moment necessary for you, please rest and then make a pause and then continue to work. Also, you can ask me questions in the general chat of the floor. I will try to answer them. So let's start. Today we are talking about very specific, very interesting organ with the features of metabolism, with the many functions. So let's talk about liver. The liver is a cone-like shaped dark reddish brown organ that is located in the upper part of the hand-right abdominal cavity, under the diaphragm and near the stomach, hand of pancreas, kidney and intestine. To supply blood to the liver, there are two distinct sources. Oxygenated blood flows in from the hepatic artery. Nutrient-rich blood flows in from the hepatic portal vein. The liver holds about 13% of the body's blood supplied at any given moment. All the blood from stomach and intestines passes through the liver. More than 500 vital functions have been identified with the liver. Some of more well-known functions include production of bile that will help for digestion of fats in the small intestine, conversion of excess of glucose to glycogen for storage and maintenance of glucose level in the blood, regulation of blood level of amino acids, which are building blocks of proteins, production of certain proteins for blood plasma, regulating blood clotting, and resisting infections by making immune factors or removing toxins. Production of triacylglycerols, cholesterols, phospholipids, and also proteins for their transport. Conversion of poisonous to less toxic compounds, detoxification of ammonia by synthesis of urea, metabolism of nucleotides, clearance of bilirubin, clearance of blood from drugs, and then. And knowledge about specific structure of liver helps to understand this polyfunctional character of this organ and some features of metabolism of substances. The functional part of liver tissues, parenchyma, is made up of around 80% of hepatocytes. The term parenchyma is a Latin word from Greek that means visceral flesh. Ancient anatomists used it referring to functional parts of organs, in contrast to term stroma, which refers to the structural tissues of organs that are usually made from connective tissue. Other cells of liver are represented by endothelial sites, of course, different satellite sites, and dendritic cells, macrophages, Kupffer cells, and then. The pyramidal section of the liver segment, is in connection with so-called hepatic triad, the branch of portal vein of the second order, its own branch of hepatic artery, and corresponding branch of the hepatic duct. Each 8 liver segments consist of 1,000 lobules, small lobes. These lobules are connected with small ducts while canaliculi, and then they connect with the larger ducts to form the common hepatic duct. The common hepatic duct transports the bile made by liver cells to the gallbladder, and then to the duodenum, first part of intestinum, via the common bile duct. Let's summarize our knowledge about functions of liver. This is, of course, participation in digestion and distribution of nutrients. It's a participation in all types of metabolism: proteins, lipids, carbohydrates, vitamins, minerals, nucleic acids, excretory function, destruction of toxic substances, both formed in body or foreign, and depot, of course, of blood, glycogen, iron, vitamins, etc. Look to the composition of bile. The greatest quantity is represented by bile acids, but of course, put attention that phospholipids are also presented with significant quantities. Minor components are cholesterol, bilirubin, proteins, and inorganic compounds. It's necessary to tell that when bile acids are secreted to the intestinum, they can be reabsorbed and through the portal vein, through the portal system, return to the bile and be secreted again. It is named enterohepatic circulation, and each molecule of bile acid can circulate 7-10 times, but after this, of course, they will be excreted with the feces. Bile acids are synthesized in liver from cholesterol, and our basic primary bile acid is cholic acid. After this, in liver, it can be conjugated with glycine and taurine. I remind you that taurine is also formed from amino acid cysteine to form glycocholic acid and taurocholic acids. After secretion, all of these acids, cholic can be converted to deoxycholic acid, conjugated to glycodeoxycholic acid and taurodeoxycholic acids under the action of microorganisms in our intestine. So let's summarize the role of bile acids secreted to the intestines. This is to emulsify fats, facilitating the action of lipase. It's to activate pancreatic lipase. It's to participate in the absorption of fatty acids, monoacids, glycerols, and cholesterol by forming with them hydrophilic complexes, micelles. Bile acids also activate the motility of small intestine, stimulate the production of mucus and gastrointestinal hormones such as cholecystokinin and secretin. They prevent the adhesion of bacteria or protein segregates due to interaction with biliary tract epithelial cells. The bile ducts of healthy people are sterile. Bile activates also kinasogen, turning it into enterokinase, which can activate trypsinogen, converting it to trypsin. So, That is, bile acids also necessary for the digestion of proteins. They help in this process. Let's discuss metabolism of carbohydrates in liver. Of course, this is glucose oxidation with energy production that in liver will be done by aerobic pathway, including three main stages: glycolysis, conversion of pyruvate to acetyl-CoA, and then citric acid cycle. I allow myself to remind you the general patterns of energy production in our body. At first, complex molecules, polymers, are coming to our gastrointestinal tract, where they will be digested to more smaller molecules, and these small substrates will be absorbed and transported by blood to all of the cells. Intracellular, they will be involved into the reactions of catabolism. Not all of them will be oxidative reaction, but we will put attention to them now. Part of reactions are oxidative, when we are taking electrons from the substrates, and it will lead to the formation of reduced coenzymes of dehydrogenases, NADH2, FADH2, which after this will be involved to the electron transport chain, to the final acceptor of the electrons, to oxygen, and then chemiosmotic potential will be formed, ATP will be synthesized. All of the process is named oxidative phosphorylation. There is only one other pathway how to obtain ATP in our body. These are reactions with macroergic substrates. It means with substrates that already contains macroergic bond. And if we have specific to these substrates transferase, with the help of this enzyme, we can obtain ATP directly near the substrate. Due to this, such a phosphorylation is named substrative phosphorylation or substrate level phosphorylation. So we can obtain ATP by oxidative phosphorylation or substrative phosphorylation. In aerobic oxidation of glucose, we can separate three stages. This is aerobic glycolysis, where one glucose is converted to 2 pyruvic acids with production of 6 or 8 ATPs. 6 or 8, it depends from the type of shuttle mechanisms, because reduced coenzyme NADH2 is formed in cytosol during the glycolysis. In liver, note that we have glycerol phosphate shuttle mechanism working, and due to this 6 ATP we can produce in liver during the glycolysis. Then the second stage, my dears, very often in your answers you are forgetting about this very important stage. The quantity of produced energy is comparable with the glycolytic energy production. So oxidative decarboxylation of pyruvate or pyruvate dehydrogenatic reaction. In this reaction, 2 molecules of pyruvates are converted to 2 molecules of acetyl-CoA. It means activated 2-carbon unit is formed to come after this for citric acid cycle. Citric acid cycle is started from the reaction between acetyl-CoA and oxaloacetate with formation of citrate. My dears, between glycolysis and citric acid cycle, don't forget about pyruvate decarboxylation. And decarboxylation also, 2 molecules of carbon dioxide here at this stage go to the air. Third stage is a Krebs cycle, citric acid cycle, where 2 acetyl coenzyme A completely catabolized and 4 molecules of carbon dioxide go away. Look, that this is the more rich with energy stage. It is possible to obtain 24 ATP molecules here. Very often you tell us during your answers that after this respiratory chain starts to work. It's not completely correct. All 3 stages are combined with respiratory chain, because in all of 3 these stages reduced coenzymes of dehydrogenases are formed. So from aerobic glycolysis, from oxidative decarboxylation of pyruvate, from citric acid cycle, reduced coenzymes will bring electrons to the electron transport chain to oxygen with production of ATP. So all 3 stages contain reactions of dehydrogenation. Dehydrogenation is equivalent of oxidation in our body. So In addition to oxidation of glucose, of course, metabolism of fructose and galactose can be performed in liver. And you remember that enzyme apathias of these pathways can cause diseases like galactosemia, fructose intolerance, and then gluconeogenesis, the synthesis of glucose from non-carbohydrate products, also very active, is performed by liver. And this is the second pathway, how to obtain glucose during the starvation periods. Pentose phosphate pathway. Put attention that this pathway is active in liver. Compare, only 0.3% of glucose can be converted by this pathway in muscles. In liver, nearly 3% of glucose will be oxidized by this alternative oxidative pathway to produce NADPH2, which is necessary for lipid synthesis and antioxidant reactions, and with the production of ribose 5-phosphate for nucleic acids and nucleotides metabolism. Synthesis and degradation of glycogen, of course, the important processes that are performed in liver, and integration between gluconeogenesis and glycogenolysis in liver is named Cori cycle. All of this process allow liver to do its main function in carbohydrate metabolism. Remember this, that the main function is to maintain the stable glucose concentration in the blood. The first reaction that can be done in any cell with glucose and in liver cell the same, this is phosphorylation of glucose and conversion it to glucose 6-phosphate. Do you remember? In liver, for this reaction, we have isozyme of hexokinase enzyme, which is named glucokinase, and it differs from the hexokinase by the several properties. You can see on the graph differences in the work of glucokinase and hexokinase. The main differences includes difference in Michaelis constant. Put attention that Michaelis constant of glucokinase is near 10 millimole per liter. I remind you that Michaelis constant is equal to the substrate concentration at which velocity reached half of maximum. So glucokinase will be active only at periods where concentration of glucose in portal vein will be higher than at norm. At norm, you know, it is near the highest value is near 5.5 millimoles per liter. The Michaelis constant of hexokinase is significantly low. It's nearly 0.1 millimole per liter, and this constant of Michaelis of hexokinase allows for peripheral cells to take glucose even during the starvation between meals and then. Also important that glucokinase is not inhibited by glucose 6-phosphate. The concentration of glucose 6-phosphate must be high in the liver cells because glucose 6-phosphate is used in liver cells not only to be oxidized but also for the conversion of glycogen. And hexokinase, which is inhibited by glucose 6-phosphate, will regulate the uptake of glucose by other cells at the level which is necessary only to their survival to normal rate of metabolism. So glucose, after conversion to glucose 6-phosphate, after this can be directed to oxidative pathway or can be used for glycogen synthesis or for pentose phosphate pathway. Let's look what enzymes will determine the velocity of all of these metabolic ways. The first we named already, this is glucokinase that convert glucose to glucose 6-phosphate. Then, if phosphofructokinase and pyruvate kinase will work, aerobic oxidation will be started. These are two more regulative enzymes of aerobic glycolysis. Then pyruvate is converted to acetyl-CoA and go to citric acid cycle. There are several regulative enzymes for citric acid cycle, but the most important is isocitrate dehydrogenase. In excess of glucose 6-phosphate, it is converted to glucose 1-phosphate by mutase, and then glycogen synthase uses it for the glycogen synthesis. If necessary, to put glucose to the blood, glycogen phosphorylase will convert it back to the glucose 1-phosphate and then to the glucose 6-phosphate. Here we need special enzyme for dephosphorylation of glucose 6-phosphate and conversion it to free glucose. Only free glucose can leave the cells. Phosphorylated monosaccharides cannot leave the cells. So we need enzyme which is named glucose 6-phosphatase. Another pathway to obtain glucose, this is process of gluconeogenesis from non-carbohydrate products that can be converted to pyruvate and then to oxaloacetate, because we cannot obtain phosphoenolpyruvate from pyruvate directly. The last reaction of glycolysis is irreversible. Non-carbohydrated products like lactic acid, like glucogenic amino acids and glycerol can be converted to pyruvate, then to oxaloacetate with the participation of pyruvate carboxylase. Elongation of pyruvate and synthesis of oxaloacetate is catalyzed by this enzyme. And finally, with the help of glucose 6-phosphate dehydrogenase, glucose 6-phosphate can be converted to 6-phosphogluconate, 6-phosphogluconic acid, and go to pentose phosphate pathway that, in addition to ribose 5-phosphate, can produce big quantity of reduced NADP, NADPH2, for lipid synthesis in liver and for participation in antioxidant reactions to protect the lipids of membranes. Look to the enzymes involved into the synthesis of glycogen in the liver. In addition to named glucokinase, we need in mutase to convert glucose 6-phosphate to 1-phosphate and glucose 1-phosphate uridyl transferase that will activate our glucose before incorporation of this residue to the glycogen and elongation of glycogen. The most important enzyme is, of course, glycogen synthase, but also we need in branching enzymes that will transfer small parts of the linear part to form 1,6 alpha glycosidic bond between the branches of glycogen. You know that glycogen is a branching homopolysaccharide. Degradation of glycogen is controlled by phosphorylase. Phosphorylase is named phosphorylase because cut glucose in a view of phosphate with participation of phosphoric acid. It's similar to the formation of name of the enzymes hydrolases. It's a cleavage of bond with attachment of water. Then by mutase, it is converted to 6-phosphate, and we named already this enzyme glucose 6-phosphatase is necessary to cut phosphate group. Put attention that this enzyme is absent in muscles. It is not expressed in muscles, and due to this, glycogen, which is synthesized in muscles, cannot be used to maintain level of glucose in blood. Total quantity of glycogen is higher in muscles because mass of muscles is higher than mass of liver, of course. But only liver participating in the maintenance of blood level of glucose. Muscles are using glycogen or only for themselves to obtain energy during the contraction. In deficiency of these enzymes in liver, who remember, the special glycogenosis can be developed, and the name of this disease is von Gierke disease, deficiency of glucose 6-phosphatase. The symptoms will be long periods of hypoglycemia without uptake of food, and of course this is an hereditary disease, and symptoms are started from the burning. It's a slow growth, slow development, mental retardation, and sometimes even death. relationships between glycogenolysis in muscles during their contractions and gluconeogenesis in liver is named Cori cycle, named after authors who described it and obtained Nobel Prize for the works about glycogen and enzymes. Glycogen in muscles can be used as a source of energy, and during the anaerobic glycolysis, the great quantity of lactate can be produced. The fate of this lactate is to be used for gluconeogenesis, but the most high potential for gluconeogenesis, of course, is in liver. So lactate is produced by blood to the liver, where it is used for the synthesis of glucose in the process of which is named gluconeogenesis. During the period of relaxation of muscles, it means, of course. Then this glucose can be used both for synthesis of glycogen in liver or go with the blood return to the muscles where it can be used for the synthesis of glycogen and then for oxidation. So these relationships between glycogenolysis in muscles and gluconeogenesis in liver is named Cori cycle or glucose lactate cycle. The most important function of liver, we told this, is to maintain the stable glucose level in blood, and we need to mention that the most important two hormones that regulate this process, these are insulin and glucagon, two pancreatic hormones. Look and put attention once more that glucagon will stimulate glucose synthesis and export to the blood. So degradation of glycogen and gluconeogenesis is under the action of glucagon. Glucose go away. It's a memory rule to remember what is the function of glucagon in carbohydrate metabolism. Insulin, opposite, will stimulate usage of glucose for oxidation and for synthesis of glycogen in the liver. So insulin for other tissues, it's glucose go inside of the cells, and inside of the cells, please go somewhere to oxidation or to glycogen. Insulin and glucagon helps to the liver to maintain level of glucose and supply all peripheral tissues and especially brain with glucose during the starvation period and between meals. Let's discuss about metabolism of lipids in the liver. Of course, lipolysis can be done in liver. Lipolysis stages, these are fatty acids, beta oxidation, conversion them to acetyl-CoA, and then oxidation of acetyl-CoA in Krebs cycle, and oxidation of glycerol. If you remember, glycerol must be phosphorylated, and after dehydrogenation, it can be incorporated into the glycolytic pathway. But more characteristic for liver anabolism of lipids. Why? Because usual fuel for us is glucose, and of course liver is taking glucose primary in our body by the portal vein after absorption in the intestine. Synthesis of triacylglycerols and phospholipids, important function of liver. It includes the synthesis of fatty acids after carboxylation of acetyl-CoA and formation of malonyl-CoA. It is used for the synthesis of palmitate. Then palmitic acid, if necessary, can be elongated, desaturated. This will be performed by microsomal enzymes. And then phosphorylated glycerols, activated glycerols, participate with the fatty acids and acyl coenzymes A transferase in the formation of subsequently phosphatidic acids and then phospholipids or triacylglycerols. Of course, liver cannot store the lipids. We mentioned polyfunctional character of this organ. There is no place for storage of liver of lipids in the liver because many enzymes must work with a high velocity. Other functions must be performed. So liver is actively synthesizing lipids, but must also organize their transport from the liver to other peripheral cells and to adipose tissue where, for example, triacylglycerols can be stored. So liver must synthesize apolipoproteins, so special proteins to cover hydrophobic lipids to make them more soluble in blood water stream, and this very important function we will a little bit detailable look a little bit later. One more, of course, synthesis of cholesterol for membranes and formation of bile acids directly in the liver, and ketone bodies formation. I will remind you that ketone bodies, these are water-soluble compounds like beta-hydroxybutyrate and acetoacetate that are formed from acetyl-CoA. In excess of this acetyl-CoA, in small quantity, liver normally is synthesizing ketone bodies to supply our peripheral organs, especially cardiac muscles, skeletal muscles, brain. Brain is not doing beta oxidation, but can use ketone bodies with the water-soluble form of acetyl-CoA. It's more convenient to give acetyl-CoA for the peripheral cells than glucose, because glucose must be converted to acetyl-CoA by many reactions. But it is not possible to do this all the time for liver. Because ketone bodies can change the pH of blood, you must understand this, and during the oxidation of fatty acids, a big quantity of acidic components can be formed. Due to this, we prefer use glucose normally, which is water soluble in comparison with lipids, and ketone bodies are synthesized but in small quantity. Quantity of ketone bodies can be increased only in case when glucose is not available for oxidation. This is during the starvation, long starvation, and this is during the diseases accompanied with the absence of insulin, like diabetes mellitus. But as I told you, one of the most important function of liver in lipid metabolism, this is the synthesis and formation of lipoprotein particles. I want to remind you what are them, how do they constructed. In all lipoprotein particles in general, we have hydrophobic nucleus that is usually formed by triacylglycerols, absolutely hydrophobic lipids, and free cholesterol. Free cholesterol cholesterol esters. Free cholesterol can participate in the formation of hydrophilic cover of the lipoproteins. Here we can see phospholipids, which are amphiphilic, and due to this can form special cover for the hydrophobic nucleus. And of course, proteins. Proteins are water soluble, and due to this, they will help for all the particles to be more soluble in the blood. And you remember that we are dividing all lipoproteins in different groups, independence from the quantity of proteins that are present in them and composition of lipids. The highest type of lipoproteins, I remind you, is formed in intestinum. These are chylomicrons that are formed by intestinal cells, and they contain biggest quantity of triacylglycerols that are resynthesized in enterocytes after the absorption of lipids from the gastrointestinal tract. Remnant chylomicrons after transportation of these triacylglycerols to peripheral tissues will be taken by liver, and all other types of lipoproteins are formed in liver. Liver is changing the composition of lipoproteins, and they obtain more and more proteins, and composition of lipids is changed. You see that the quantity of phospholipids is increased, quantity of cholesterol is increased, and finally the quantity of protein is significantly increased in the high density lipoproteins. They are not heterogenic, as you know, and very low density and low density lipoproteins are heterogenic. So function of liver to synthesize apoproteins and to create correct balance between the components, lipid components of lipoproteins. This is a principal question in the formation of such a disease like atherosclerosis and different types of hyperlipidemia. Okay, summarize the participation of liver in the protein metabolism. This, of course, protein decomposition and interconversion of amino acids by different reactions that can be done in liver, especially these are reactions of transdeamination. It's a problem that direct deamination we can do only for glutamic acid because other oxidases of L-amino acids, usually FMN and FAD containing, are not working actively. Their optimum of pH is near 10 and they are working very bad. So what to do? We have glutamate dehydrogenase that can easily deaminate glutamate, and all of the amino acids can be easily transaminated at first with alpha-ketoglutarate, which is absolutely normal component of the hepatocytes, and only after this glutamate will be deaminated and ammonia will be taken away. Reactions of transaminases also are very necessary for the synthesis of non-essential amino acids, which after this will be used for the synthesis of proteins. Conversion of amino acids to low molecular weight nitrogen-containing compounds. Liver is the center of the production of many very important substances which are still nitrogen-containing. These are, for example, already mentioned today, taurine for the creation of taurocolic acid. It also can be first stage of creatine formation. This is purine synthesis, and then. Conversion of proteins into the carbohydrates and lipids. This integration between the all types of metabolism very good is visible, especially in the action of liver. As we told, glycerol from the lipids can be incorporated into the processes of gluconeogenesis. Also, glucogenic amino acids from the proteins can be incorporated in this moment. In its order, for example, pyruvate obtained from glucose can be used by reactions of transaminases for the synthesis of alanine if you did not obtain alanine. It's a bad example, but because alanine is widely distributed in the dietary proteins. But anyway, it's a possibility to understand that we can redirect carbon from the one type of organic compounds to another one. What we cannot synthesize, remember, we cannot synthesize glucose from acetyl-CoA, unfortunately, and we cannot synthesize so-called essential factors of the food. These are polyunsaturated fatty acids from the lipids, and these are essential amino acids. For the protein, it's principal because nearly half of amino acids are essential. Protein synthesis, of course. With the high velocity, proteins are synthesized in liver because these are blood plasma, of course, these are enzymes of liver's own enzymes, and of course, these are blood plasma proteins, albumin, globulins. Between globulins, it is necessary to mention blood clotting proteins. Diseases of liver very often are accompanied by the disorder of blood coagulation. And detoxification of ammonia, urea synthesis. This process absolutely unique for the liver, and uremia can be caused by the hyperproduction of urea, of course, in rare cases, but more often is created by the kidney disease and absence of possibility to filtrate urea to the urine. Ammoniemia, this is the absolute sign of liver disease, because if ammonia is not detoxified, it's a sign of problem with the factory where it is normally synthesized. So look to this unique for liver process, urea synthesis, which is partly will be done in mitochondria, partly in cytosol of liver sites, liver cells. Ammonia, so-called waste nitrogen, by the special enzyme activity of which is most important for the velocity of all the process, and is regulated by the protein-rich diet, convert ammonia and free ammonia and carbon dioxide to the substance which is named carbamoyl phosphate. Then the special enzymes converted it subsequently from ornithine to citrulline. Then for the arginine succinate, looked at second molecule, second atom of nitrogen can be brought by aspartate, and fumarate that is formed after this can go to the citric acid cycle, you remember. Then arginine is formed, and the special only for the liver tissue enzyme arginase will convert arginine back to ornithine, which can go to the cycle back, and urea. Urea is transported by blood to the kidney, where it is filtrated in urine. The most important source of free ammonia in the liver, this is, as we told already, transdeamination of amino acids. I remind you, these are subsequent reactions of all amino acids with alpha-ketoglutarate, with the formation of glutamic acid and corresponding to the amino acids ketone acids. Ketone acids, so carbon skeletons without nitrogen, after this can be used for the catabolism, for example, can be incorporated into the oxidative processes, or sometimes are used for the synthesis of important substances. Then formatted glutamic acid is converted to alpha-ketoglutarate back by the enzyme glucose 6 glutamate dehydrogenase. Free ammonia used by urea cycle for the synthesis of urea. Alpha-ketoglutarate can go to citric acid cycle or to the next reactions of transamination. This process we are naming indirect deamination of amino acids or transdeamination. For the synthesis of urea, not small quantity of ATP is necessary. You remember this. And what is the most important source of ATP in our body? Yes, this is the Krebs cycle or citric acid cycle, which is also described by Krebs, same like urea cycle a little bit earlier. So all these two cycles are strongly related one to another, strongly bound one to another, and determine the velocity and effectiveness of our metabolism acting. Due to this, very often you can meet the characteristic of these processes like like a mentioning of bicycle of Krebs. You can see that there is a connection between these two cycles. This is energy, this is aspartate that can be formed from the oxaloacetate, and this is also fumarate that can go back from the ornithine cycle to the citric acid cycle. As quickly as we are moving with this Krebs bicycle, as quickly we are leaving. One more cycle that is necessary to mention in characteristic of protein metabolism in liver. This is glucose-alanine cycle. Relationships between the processes of anaerobic glycolysis, glycolysis generally, transamination, and urea synthesis. In muscles, after formation of pyruvate, the produced ammonia can be incorporated into the alanine structure by reaction of transamination, for example, or usage of amino acids from the muscles. Alanine is a small amino acids, very good water soluble, that can come after this with blood, same like lactate, to the liver, where reactions of transaminations are very active, and be converted back to the pyruvate with release of ammonia. Release of ammonia in liver is not dangerous because here we have processes of urea synthesis. Pyruvate after this can be used for the glucose synthesis, and glucose, same like in Cori cycle, after this can return to the muscles. So this is so-called additional way how to transport ammonia from the peripheral tissues. It is characteristic for muscles and a little bit for enterocytes, cells of intestine. They also sometimes produce small quantity of alanine to transport its ammonia to the liver for detoxification. For other tissues, for example, for brain tissues, is more characteristic formation of glutamine and in less quantity asparagine, amides of glutamate and aspartate to transport this ammonia from the brain also to the liver for detoxification, or directly like glutamine to the kidney by the action of glutaminase. This ammonia will be revealed and excreted to the urine. Big function of liver, this is detoxification. Detoxification that can be done not only for the foreign compounds, pollutants, cancerogenic and toxic compounds, but also metabolism of a big quantity of our own compounds is related with the action of microsomal enzymes. I will remind you that there are no such organelles in the cells which will be named microsomes. Microsomes, these are particles that it is possible to obtain after degradation of endoplasmatic reticulum of the hepatocytes. If you will take hepatocytes and destroy them and centrifugate, you can obtain microsomal fraction via pieces of membranes of endoplasmatic reticulum together with enzymes which were attached for them, formed such a vesiculae that were named microsomes. Enzymes that are present on these membranes involved into the process of metabolism of our own compounds and also exchanges of toxic compounds and compounds that is difficultly to excrete from our body due to their hydrophobicity. We told with you what substance is more toxic, hydrophobic or hydrophilic. Our excretion we are doing usually with sweat, with urine, so with fluids. So as more substance is hydrophilic, as quickly we can excrete it from the body and stop its toxic action to our body. We are dividing the process of detoxification in liver to two phases. In the phase 1, preparative hydrolysis, of course, can be done, and then small reactions of modifications, including hydroxylation, amination, attachment of tier group or carboxylic group can be done. But more often, these are a reaction of hydroxylation. It's understandable that appearance of electronegative oxygen will change the property of the substance. As a result of first phase of detoxification, usually hydrophilic properties of the substance must be a little bit increased. The most important enzyme that is involved into the hydroxylation reaction, this is cytochrome P450. And due to the function of liver, detoxification is mainly a function of liver. The highest concentration of this enzyme you can find, of course, in microsomes, in hepatocytes, in the plasmatic reticulum of liver cells. Cytochrome P450, this is the heme-containing enzymes with high, with very wide substrate specificity, and it catalyzes the incorporation of one atom of oxygen into the substrate. So according to the mechanism of action, this enzyme is monooxygenase. Of course, the second atom of oxygen must be neutralized. It will be converted to water, and due to this monooxygenase system, all the time are working with dehydrogenases. Don't forget about this. The second atom is usually converted to water. Here on this slide, you can see the examples of reactions that are catalyzed by the cytochrome with the participation of cytochrome P450, and it's not only drug conversion to the substances that can be after this easily excreted from the body. This is also reactions of modification of our own products. These can be fatty acids, these are steroid hormones modifications that can be also with high velocity done in the cortex of suprarenal glands, of course. And not only hydroxylation reactions are, not only cytochrome P450 is used for reactions of hydroxylation, you know, also very well known for your reactions, is a conversion of phenylalanine to tyrosine. Differences on the presence of one OH group. These are also example of reactions that are performed by microsomal enzymes of liver. The phase 2 includes conjugation reaction. It means attachment to the compound, especially for the excretion, something which is very hydrophilic and increase hydrophility of the substance significantly. What reactions can be used? This is glucuronidation, attachment of glucuronic acid. Glucuronic acid is a derivative of glucose, and you know very good the substance which is detoxified by glucuronidation. This is the bilirubin, resulting substance from heme catabolism. But also other reactions can be done. This is attachment of sulfuric acid residues, acetylation, methylation, conjugation with the tripeptide glutathione, amino acids like glycine, glutamic acid, taurine, residue of cysteine. And then the result of second phase is the remarkable increase of hydrophilic properties of the substances, and this allow for the liver to excrete these substances by production is to the bile, secretion is to the bile, and through the bowel with feces, or put this substance in not very high concentration in the blood, which will be filtrated in kidney, and then the substance will be excreted with urine. Don't forget about participation of liver in pigment metabolism. In our red blood cells, we have a hemoglobin, and hemoglobin, it's a complex protein that contains heme. Same with cytochrome P450, catalase, peroxidase, and other enzymes. Heme is interesting compound, and as in some other cases, this compound can be synthesized in our body, but cannot be completely catabolized for CO2 and water. After destruction, after hemolysis of the old erythrocytes, hemoglobin is coming to the cells of reticuloendothelial system, where it is converted to substance which is named bilirubin through some stages. Bilirubin is compound toxic, non-soluble. It must be transported from reticuloendothelial cells of all of the body to the liver for its detoxification, and it can be done in the complex with albumin. This complex of lipid-soluble, water-insoluble bilirubin that is formed from inside of reticuloendothelial system is named indirect bilirubin or unconjugated or free bilirubin. It is not free from albumin, but it is free from glucuronic acid still. It is indirect because we cannot do the direct reaction to find and to estimate its quantity. At first, we need to precipitate proteins. So due to this indirect reaction, this bilirubin is named indirect. Unconjugated bilirubin with the help of albumin is transported to liver, where it is converted to diglucuronide. With the help of the special enzyme glucuronyl transferase, you know this already. And this bilirubin, due to the specific structure of liver, I show you this and remind you this about triads and about the special structure of liver, partly can also come to the blood. Due to this, we have both fractions of bilirubin, indirect and direct, in our blood normally. The quantity of these fractions of bilirubin and their balance can be changed in case of jaundices, yes, in case of excessive hemolysis or inflammation of parenchyma cells of liver, or obstruction of bile ducts. Three types of jaundices we discussed already during the classes. Then bilirubin is excreted together with the bile to the intestinum, where it is converted to some colorless pigments. Colorless pigments is interesting, but you must know and remember that urobilinogen or mesobilinogen, which is formed in the middle parts of intestine, it is really colorless, so the name pigment is very strange for it. It's a problem of nomenclature. I told you about enterohepatic circulation, so the part of urobilinogen already converted bilirubin to it can be reabsorbed in the intestine and returned to the liver. Normally working liver is not allowed to urobilinogen go to the general circulation. It must be cut to monoendoperols by very good working liver. So only part of urobilinogen can be transported by intestinum till the rectum and then converted to stercobilinogen, and stercobilin, it's a normal pigment of our feces, it is brown, and normal color of feces is brown. But in the lowest part of rectum there is a so-called portocaval anastomosis. It's anastomosis between portal system and system of vena cava inferior, and part of stercobilinogen can be absorbed at this moment and go to the general circulation. It will go without destruction to the kidney, where it is filtrated and gives the light yellow color to our urine due to the presence in very small quantities. So the pigment of feces and urine is the same. This is stercobilin. Urobilinogen, real urobilinogen, mesobilinogen, from the middle part of intestine can go to the urine only in pathological states where liver is not normally working. It is characteristic mainly for the situation with parenchymatous jaundice with hepatitis, more often viral or sometimes caused by chemicals. Very short about vitamin metabolism in liver. We need to mention that, for example, active form of vitamin D must be formed in liver. These are also reactions of hydroxylation. We talked about this type of this reaction in liver. The precursor of vitamin D is coming to the body or synthesized in our skin under the action of ultraviolet light. But after this, first time in kidney and then in liver, or at positions of 21, 1 and 24, they must be hydroxylated to be calcitriol. It means 3 hydroxyl group must be formed in them to be activated and to be converted to active vitamin D. So liver is participated. Also, formation of vitamin A from the precursors, from the carotenoids, also can be done in liver. Depot of B12 and a little bit folic acid can be formed in liver because cyanocobalamin, this is water-soluble vitamin from the water-soluble vitamins group, but it is really very not very good water-soluble compounds. We have special transporter proteins for them, and also liver can serve as a depot of this vitamin. Due to this development of deficiency of B12 usually is not immediately after formation of the conditions for this. For example, in case of gastrectomy, surgical operation where all the stomach will be taken away and intrinsic factor of Castle will not be produced, dietary B12 cannot be absorbed. But after gastrectomy, pernicious anemia can develop after 1 or 2 years because in our liver we have a depot of B12. Other vitamins can be also stored, like fat-soluble vitamin E. And what is important, put attention that active forms of for the active coenzyme forms for the vitamins from the group B also require different changes. For example, for B1, thymine must be converted to TDP, so it must be phosphorylated, and these reactions are also performed in liver. I hope that you understood that according to all of this function, liver is possible to name our metabolic heart. It's my opinion. You should agree with me, I think. And thank you for your attention. Continue to study and stay safe. Goodbye.